severe cataplexy than male dogs. However, this sex difference disappeared once the dogs reached adult age, 30. Additionally, female hypocretin knockout narcoleptic mice showed more frequent cataplexy episodes than male mice, and these episodes were influenced by the estrous cycle, suggesting that hormonal factors might modulate disease phenotype, 32. The clinical presentation of narcolepsy in the affected Dogo Argentinos was consistent with previous descriptions of the disorder in other breeds. Owners reported a decrease in episode frequency over time, which could indicate an adaptive mechanism. In humans, it has been reported that narcoleptic children have a higher frequency of cataplexy events, 33. One study also showed that in young dogs, the severity of cataplexy tends to worsen until it reaches a plateau at 16 to 24 weeks, 30. Two of the dogs in this study were treated with venlafaxine. However, the owners did not report any improvement in clinical signs. This aligns with the literature, which reports that treatment options for narcolepsy in dogs are usually insufficient to fully control narcolepsy, 34. Specifically, reports of venlafaxine use in dogs with narcolepsy identify questionable efficacy, 35, 36. This genetic discovery and familial investigation has several limitations. A complete blood cell count and a biochemistry panel were not performed in all the affected dogs of this study, so it is unknown if other potential diseases or phenotypes were present. However, the narcolepsy diagnosis relies primarily on the history and clinical signs, which were characteristic in all of the reported dogs. Additionally, we obtained blood samples from a few related dogs of one of the narcoleptic cases, but did not have samples from their grandsire, who we predict has a copy of the duplication based on the suspected inheritance mechanism and absence of the duplication in the grandam. We do not have DNA from the two parents of UF1 and thus we could not determine the duplication presence for either. We failed to precisely genotype the 5 dogs with the tandem duplication and rely on circumstantial evidence to propose an inheritance pattern. Additionally, the lack of tissue from affected cases limited the ability to understand the effect of the identified duplication on protein. In conclusion, we identified a novel tandem duplication in the HCRTR2 gene in Dogo Argentino dogs with clinical signs of narcolepsy, expanding our understanding of the genetic heterogeneity associated with this disease. Prevalence in the breed, detect carriers of the duplication, and implement preventive measures to mitigate the spread of familial narcolepsy within the breed. Testing for the presence or absence of the duplication is immediately available, and the development of a qPCR test to detect zygosity will be a valuable addition. SVs can be categorized into duplications, deletions, insertions, inversions, translocations, and copy number variation, CNV, and can have a range of effects from advantageous to deleterious, 24, 25. Several phenotypes within dogs have been attributed to SVs. For example, retrogene insertions of FGF4 are causal to chondrodysplasia and chondrodystrophy, 26, and red coloration in poodles is linked to an SNN retrogene insertion upstream of GPR22, 27. Furthermore, a 400 kb tandem duplication within the Duchenne muscular dystrophy, DMD, gene was linked to muscular dystrophy in Labrador retrievers, 28. Unfortunately, SVs are notoriously difficult to discover in short-read WGS data, 29, and in many cases, SVs are discovered through visual inspection of mapped reads. In our case, we relied on SMOOV and Lumpy to discover and genotype SVs in the narcoleptic Dogo Argentinos and a wide collection of unaffected dogs to facilitate discovery. The outcome highlights the importance of considering and annotating SVs while searching for genetic causes of disease phenotypes. While gene duplication can lead to increased copy numbers, facilitating the development of new gene functions and the formation of gene families, the tandem duplication observed here was only partial in nature, resulting in only some exons being duplicated. In this case, exons 3 and 4 were duplicated. Due to a lack of RNA, we were unable to determine if these additional exons are included in the mature transcript or if their presence changes splicing patterns and introduces premature stop codons or changes the coding sequence in another deleterious manner. However, if the additional exons are included in the mature transcript, then the number of transmembrane domains increases and likely affects how the protein embeds into the cell membrane and/or communicates external information into the cell. Although we cannot thoroughly investigate the molecular consequences, this duplication is most likely responsible for the narcoleptic phenotype, but future investigation should aim to explore the structural and functional impacts of the tandem duplication within the HCRTR2 protein. All the dogs evaluated in this study were males. However, it was reported that one of the dog's littermates, a female, was also showing similar clinical signs. A definitive diagnosis of the underlying cause of the seizures was not obtained, and we were unable to access her medical records. This raises the question of potential sex differences in the expression and severity of the clinical signs caused by this specific duplication. In narcoleptic Dobermans, it has been shown that female dogs show earlier structural variant validation and segregation. We designed primers to amplify and isolate the 5-foot and 3 bends, and the junction of the identified tandem duplication, figure 4. Products were successfully generated for the 5-foot and 3 bends in all Dogo Argentinos, table 1, and a control boxer, figure 4. However, primer combinations designed to amplify the junction only yielded product in the 3 narcoleptic dogs whose NM1 is silent, figure 4C. By mapping duplication boundary and junction sequences to the reference genome, we determined the beginning of the second duplication was identical to the sequence starting CHR12:228244096, figure 4-8C, and the end of the first duplication matched sequences ending at CHR12:2284367, figure 4-B-C. Therefore, we narrowed the duplication boundaries to CHR12:2282409622843,670 NC_0492333.1 G.228240962284367 of the reference genome, covering 19,575 pp. Unfortunately, genotypes are inconclusive for PCR-positive samples in figure 4C, and DNA was exhausted before we could accurately genotype samples with dPCR or qPCR techniques. However, narcolepsy-linked mutations exhibit a recessive inheritance pattern in other breeds, and the 2x coverage increase within the duplicated region in NM1 and NM2 suggests a homozygous genotype. Since the sire and dam both had at least one copy of the duplication and were reported to be free of clinical signs of narcolepsy, we hypothesized they were heterozygous for the duplication, and the narcolepsy phenotype is likely caused by the homozygous mutant genotype, but more data are needed to support this conclusion. Protein prediction. The authors were unable to obtain RNA from blood or tissue from the narcoleptic dogs of this study in order to investigate the transcription and translation alterations caused by this tandem duplication. Therefore, we constructed a coding sequence with both exons 3 and 4 repeated, NM_00100293.1, C.403_762 DUP, to consider the consequences if both exons are included in the translated protein, NP_00100293.1 tongue sticking out. THR135_GLN254 DUP, figure 5A. Including additional exons did not yield any premature stop codons in the amino acid sequence. Since HCRTR2 is a G-protein coupled receptor, we estimated the number of transmembrane domains in the wild-type and predicted mutant protein. The wild-type HCRTR2 contains 7 transmembrane domains, whereas the mutant protein was predicted to have 9 transmembrane domains, figure 5b, and the additional transmembrane domains strongly changed the mutant protein structure predicted by AlphaFold 2, figure 5c. Discussion. In this study, we identified a novel tandem duplication spanning exons 3 and 4 of the HCRTR2 gene in Dogo Argentino dogs presenting with clinical signs of narcolepsy. This finding expands understanding of the genetic basis of narcolepsy in dogs, adding Dogo Argentinos to the list of breeds with a familial form of the condition. Our findings align with previous studies that have identified genetic mutations in T. 2.4 Structural Variance Validation. PCR primers were designed to amplify the 5' end of the SV, the 3' end of the SV, and the predicted junction of the tandem duplication using primer 318, implemented in Genius Prime 2024.0.5, Table 1. All PCR reactions consisted of 35 cycles with denaturation, annealing, and expansion temperatures at 95 degrees Celsius, 63 degrees Celsius, and 72 degrees Celsius for 30 s each, respectively, using DreamTac master mix. Amplicons were visualized on a 1.5% agarose gel. PCR products were Sanger sequenced to verify they were from the target loci and identify exact SV boundaries by mapping amplicon sequences to the reference genome containing an artificial duplication based on Lumpy's annotation with Genius Prime 2024.0.5. Protein modeling. DeepTMHMM19 was used to predict the number of transmembrane domains of both the wild-type and the mutant gene sequences, and CollabFold 20, a Jupyter notebook using MSEX2 21, and AlphaFold 2 22, was used to predict the protein folding of wild-type and mutant proteins. 3. Results. 3.1 Clinical presentation. At the time of presentation, the dogs were 6, NM3, 7, NM1, and 13 months old, NM2. The owner of NM1 reported that one of his dog's littermates, a female, was also affected and provided videos corroborating the clinical signs. All owners reported that the clinical signs had begun when the dogs were between 2 and 4 months old. Abnormalities were not detected on physical and neurological examinations in all 3 dogs, except for one dog, NM3, that exhibited sleep attacks and narcolepsy episodes during the examination when food was administered or while playing. In dog NM2, a complete blood count was performed, which revealed a mild leukocytosis with neutrophilia, white blood cell count, 25,400 cells/mm3, reference interval, 6,000 to 17,000 cells/mm3, neutrophils, 81%, reference interval, 60% to 77%, and mild normochromic, normocytic, non-regenerative anemia, HCT, 35. 9%, reference interval, 37% to 55%. Abnormalities were not detected on the urinalysis. Further workup was declined by the owners of the remaining 2 dogs. There was no reported response to venlafaxine in the 2 dogs treated, and the owner discontinued treatment after a few months. Approximately 5 years after initial presentation, 2 of the dogs in this study, NM1 and NM2, remain alive and are reported to have continued episodes. However, the owners report that the frequency has decreased over time. The 3rd male was lost to follow-up 6 months after initial presentation. At that time, he continued to have episodes. 3.2 Variant discovery. Whole genome sequencing for 2 cases and 2,766 relative controls was obtained and confirmed to represent an average sequencing depth of 26x. A total of 1927 SNVs and 2021 indels met filtering criteria, but none were found within the HCRTR2 gene, 21, Table S2. 97 SVs, 96 deletions and 1 duplication also met the filtering criteria, Table S3. The single duplication was a predicted 19,578 BP duplication on C. Familial narcolepsy in Dogo Argentino dogs is caused by a tandem duplication mutation in HCRTR2. Introduction. Narcolepsy is a sleep disorder that affects several species of mammals, including dogs, horses, cattle, cats, and humans. 1, 2. This condition is characterized by daytime sleepiness, sudden bouts of sleep, disrupted sleep patterns, quicker onset of REM sleep, and cataplexy, which involves sudden episodes of weakened or absent muscle tone. It is caused by abnormalities in the hypocretinergic, also known as orexinergic, system. Hypocretins, HCRTs, are excitatory neuropeptides synthesized by hypocretinergic neurons located in the posterolateral hypothalamus. These neurons project diffusely throughout the brain to many wakefulness-promoting areas. 3. Their primary function is to promote alertness, but they also play roles in regulating the stress response, appetite, metabolism, autonomic functions, and feelings of reward. 4, 5, 6. There are two types of HCRTs, HCRT1 and HCRT2, and they bind to two types of receptors, HCRT receptor type 1, HCRTR1, and type 2, HCRTR2. In dogs, two forms of narcolepsy have been identified, a familial and a sporadic or secondary form. The familial form is caused by a mutation in the HCRTR2 gene, documented in Labrador retrievers, dachshunds, and Doberman pinschers. 7, 8. In these breeds, the phenotype follows an autosomal recessive pattern of inheritance with complete penetrance. While the same gene is affected in all described cases, the causal mutation differs between breeds. In Doberman pinschers, the condition is caused by the insertion of a 226 base pair, BP, short interspersed element, sign, 35 BP upstream of the 5' splice donor site of the fourth exon, resulting in exon skipping and transcript truncation. In contrast, narcolepsy in Labrador retrievers is caused by a different exon skipping mutation caused by AG to a nucleotide substitution 5 BP downstream of the 3' splice site of exon 6, and an E54K missense mutation in exon 1 explains narcolepsy in dachshunds. 7. A familial form has also been described in poodles, although no causative mutation has been reported. 7. This genetic heterogeneity complicates the diagnosis of the familial form in breeds where the specific mutation has not yet been identified. The sporadic occurrence of narcolepsy has been observed in more than 17 different dog breeds as well as in mixed breeds. 9. Dogs with this form of the condition exhibit low hypocretin levels in their cerebrospinal fluid, CSF, without any alterations in HCRT2 expression. 10. Between 2019 and 2020, 3 Dogo Argentino dogs were evaluated by the Neurology Service of the Veterinary Hospital of the Universidad de la República, Uruguay, for clinical signs suggestive of narcolepsy. Additionally, one owner reported that a littermate of their dog exhibited similar signs. The occurrence of 4 cases in young dogs of the same breed, including 2 littermates, raised suspicion of familial narcolepsy. How? 2. Methods. 2.1 Dogs. Between 2019 and 2020, 3 intact male Dogo Argentino dogs, NM1, NM2, NM3, were presented to the Veterinary Hospital of the Universidad de la República, Uruguay, for evaluation due to signs of sleepiness, sleep attacks, and cataplexy. Video S1. The episodes of sleep attacks and cataplexy were exacerbated during activities such as eating, figure 1, or playing. The owner of NM1 also reported a female littermate that displayed similar clinical signs. Physical and neurological examinations were conducted on all 3 dogs. Complete blood count, CBC, biochemistry profile, and urinalysis were performed in one of the dogs, NM2. The owners declined to perform blood work in the other 2 dogs. The dogs were presumptively diagnosed with narcolepsy. NM1 and NM2 were treated with venlafaxine 2.5 mg per kilogram by mouth daily. The owners of dog NM3 refused treatment. 2 ml of ethylene diamine tetraacetic acid, EDTA. Treated blood were extracted from the affected male dogs. The female was never presented to the veterinary hospital, and samples were not obtained. The breeder of the 2 littermates, male and female, was contacted, and he consented to the collection of blood samples from additional animals in his breeding colony, which were all registered at the Uruguayan Kennel Club. Blood was collected from the sire and dam of one of the affected dogs, as well as from his maternal grandam, figure 2. Additionally, a sample was collected from an unaffected female that shared the same grandam but resulted from a unique breeding pair. In addition, samples from 3 additional unrelated, clinically normal dogs were obtained. All samples were stored at -20 degrees Celsius until processed for DNA extraction. Blood samples were obtained with owner consent in accordance with institutional practices in Uruguay. DNA extraction and sequencing. DNA extraction was performed from 200 ml of EDTA whole blood using the genomic DNA purification kit, Thermo Fisher Scientific. The genomic DNA samples were assessed for concentration and purity, 260 nanometers per 280 nanometers ratio, using a Denovix DS1 spectrophotometer, and their integrity was evaluated by 1% agarose gel electrophoresis. The whole genomes of 2 males, NM1 and NM2, diagnosed with narcolepsy were sequenced. The genome of NM3 was not sequenced because of inadequate DNA concentration. A 30 times coverage of the 2x150 BP libraries was performed on a NovaSeq X Plus system, Illumina, by Azenta Life Sciences. 2.3 Variant discovery. In addition to the 2 cases, 2,766 samples with whole genome sequencing, WGS, were included as controls. These control dogs represent a combination of samples collected for ongoing research in the NCSU Veterinary Genetics Laboratory and 1971 dogs, Table S1, from the Dog 10K Project, 11. Reads were mapped to the UU_CFAM_GSD_1.0_Rosie assembly, 12. Using the WAGS pipeline, 13. Variant calling from WGS data was performed using a standardized bioinformatics pipeline as described previously, 14. Single N. Grooming. Dogo Argentino grooming is not complicated. Because this is a short-haired dog, brushing them with a soft brush or rubber curry brush once or twice a week will keep them neat. It also helps to distribute natural oils to the entire body. The thick, white, and glossy coat has no undercoat. It sheds a little, but you won't have to deal with much doggy odor. Check the ears every week to ensure there's no dirt and debris. Ask your vet for how to clean your dogo's ears and any signs to watch out for when inspecting them. Remember to trim the nails when necessary. Feeding. As with other dogs, this breed does very well with high-quality food. This is true whether it is home-cooked or commercially available food, as long as it's nutritionally balanced canine diet. Dogo Argentino puppies will do well with puppy formula for 12 to 18 months as directed by your vet. Provide sufficient fresh drinking water and brush the teeth of your dog every day. Routine care. Different dogs require different types of care routines. While the Dogo Argentino is a low-maintenance breed, you may have to bathe them if they've been out hunting or playing in the dirt. It's an advantage that they come all white, so you'll be compelled to keep them always cleaned up. Another great trait that makes this dog lovable is that they don't drool often. So you don't have to be concerned about carrying a towel when you're not at home. Dogo Argentino possible health problems. The Dogo Argentino leads an active and healthy life. They are not prone to many dog conditions. However, you should be aware that dogos can go deaf or develop canine hip dysplasia. Deafness. Like other breeds with a white coat, dogos can develop pigment-related deafness. Some dogs will go deaf in one ear, while some in both. However, the condition is rare and can be avoided by getting puppies whose parents had normal hearing in both ears. Related, dog skin care, controlling itchy hot spots. Canine hip dysplasia, CHD. Like many other dogs, Dogo Argentinos may also develop hip dysplasia. This condition is mostly inherited. It begins as your dog is growing and causes an unstable hip joint. Exercise and watching the weight of your dog can help with the condition. Sunburn. Dogo Argentinos are sensitive to too much sun. They should, therefore, be protected from possible sunburn. To do this, take care not to let your dog stay out in the sun for long. Talk to your vet about any sun protection practices you could adopt for summer. Special considerations for Dogo Argentino. Living with a Dogo Argentino is not difficult. Schedule daily brisk walks or let them run freely in a secure area. It is also a good idea to buy a leash for this dog. They have a high prey drive and may take off quickly if they spot something they want to chase. It helps to keep your dog on a leash whenever you're walking them. Although they are not so common in the United States, Dogo Argentinos are not illegal to have. However, they may be highly gastric dilatation volvulus, GDV, also known as bloat. This condition involves the stomach twisting, which can be fatal if not treated promptly. Laryngeal paralysis. This condition affects the larynx, causing breathing difficulties. Cardiovascular disease. Heart conditions can also occur in Dogo Argentinos. Dental disease. Like many breeds, Dogo Argentinos can be susceptible to dental problems. Allergic reactions. Skin allergies and other allergic reactions can be a concern. Mast cell tumors. These are a type of skin tumor that can occur in Dogo Argentinos. Characteristics of the Dogo Argentino. Dogo Argentinos have a wide and deep-set chest that is well complemented by a muscular neck. They have a huge head that is rounded from the front to the back. The muzzle is about the same length as the skull and it concaves slightly. They also have strong jaws with teeth that meet in a scissors bite. A Dogo's nose is usually black and its eyes are dark brown, light brown, or hazel. Ears are set high, a feature that causes some owners to have their pets' ears cropped to achieve the desired pointy and triangular shape. Their thick tail is carried low and reaches the hock. Dogo Argentinos are impressively huge, with most weighing anywhere between 80 and 100 pounds. Males and females are almost the same size. A male Dogo measures 24 to 26.5 inches in height, while a female is 24 to 25.5 inches tall. Other features of the Dogo Argentino include temperament. Dogo Argentinos are protective of their home and owners. That's why they need to be socialized from an early age and get good training. Generally, a Dogo will get along with you after he's comfortable having you around. Some have, however, been known not to like other dogs. Related, what to do when your dog won't eat. Intelligence. Dogo Argentinos are smart and do well in spirited games like fetch and other dog sports that require the pulling of weights and agility. Obedience training helps to challenge the Dogo mentally. Their intelligence is evident in the many uses that people buy them for. Socialization. The Dogo is very energetic and can be difficult to control if they grow up into ill-mannered adults. They require lots of daily exercises to spend extra energy. It also helps a great deal to train them consistently as puppies and socialize with family members. Shedding. Their smooth coat doesn't shed much, so it's easy to clean and take care of. Lifespan. The average lifespan of the Dogo is 9 to 15 years. Barking. Argentinian Mastiffs are not noisy and will only bark when necessary. Cost. You can buy or adopt a Dogo Argentino for any amount between $1,000 and $4,000. The price could even go higher depending on the bloodline. Is Dogo Argentino a good family dog? Most people are concerned about big dog breeds and their ability to be good pets. In most cases, it comes down to how you train and socialize your dog. If your Dogo is well trained and socialized, it can be a good family dog. They are very Testing, a common test used to evaluate hearing in domestic animals, is recommended for all puppies, as pigment-related deafness is reported in the breed. Temperament. Loyal and loving Dogo Argentino. The Dogo is among the toughest of breeds, and it is not a breed for inexperienced or casual dog owners. It requires an experienced owner who is firm, confident, consistent, and careful, since the breed may be aggressive toward strange dogs and people and even familiar dogs. Because of its strength, the Dogo is not a good choice for children or seniors. However, it is a good watchdog and protection dog. Despite its tough nature, it is a loving, loyal, and demonstrative companion that likes close contact. Breeders suggest getting a Dogo as a puppy so that the new owner can establish a respectful relationship and initiate an obedience regime. The breed is fairly active and does best when given activities to occupy it. It is not a prolific barker. These are well-established and widely accepted generalizations about the breed. Individual dogs may differ in behavior and temperament. Dogo Argentinos are generally healthy dogs but can be prone to certain inherited conditions. Key health concerns include hip and elbow dysplasia, deafness, glaucoma, and hypothyroidism. They can also be susceptible to bloat, gastric dilatation volvulus, a serious and potentially life-threatening condition. Other potential issues include laryngeal paralysis, cardiovascular disease, dental problems, and allergic reactions. Here's a more detailed look at some of these conditions. Hip and elbow dysplasia. These are developmental conditions where the hip or elbow joints don't form correctly, leading to pain and arthritis. Deafness. Dogo Argentinos, particularly those with white coats, can be predisposed to congenital deafness. Glaucoma. This condition affects the eyes and can cause increased pressure within the eye, potentially leading to blindness. Hypothyroidism. This occurs when the thyroid gland doesn't produce enough thyroid hormone, impacting metabolism. Dogo Argentino, breed of working dog created in Argentina in the 1920s as a pack-hunting, big-game hunter. The Dogo Argentino was developed by two brothers, the Argentinian physicians Antonio and Agustin Noers Martinez, mixing the extinct old Cordoba fighting dog bred and trained to fight to the death, with several other breeds. The dog is noted for its strength and is recognized by its short and sleek white coat, colored eye patch, and powerful, muscular build. The ears are traditionally cropped in countries in which it is legal to do so. They are drop-eared when natural. Because of the Dogo's reputation as a potentially dangerous breed, it is banned, restricted from importation, and requires special dispensation in several countries, origins and history. The Mastiff-like Viejo Perro de Peli Cordobés was used for pit fighting and for bringing down large ferocious prey. However, since that breed was so aggressive that it was difficult to own, the Noers Martinez brothers mixed it with several breeds, including the Pointer to improve scenting ability, the Great Dane to increase height, the Pyrenean Mastiff for a calmer temperament, the Bull Terrier for white coat color, the Boxer for agility and personality, and the Bulldog for increased jaw strength. Later, the Dogo was the subject of a national magazine article and gained particular notice when Antonio Noers Martinez pit his Dogo against a boar and a puma, and the Dogo won. After Antonio Noers Martinez's death in 1956, Agustin Noers Martinez traveled extensively as Argentina's ambassador to Canada, allowing him to promote the breed to a global audience. The Argentine Kennel Club recognized the Dogo Argentino in 1964, the Fédération Cynologique Internationale, FCI, in 1973, and the American Kennel Club, AKC, in 2020. Introduced to the United States in 1970, it became popular with hog hunters in the western part of the country. With AKC recognition and exposure to more people, the Dogo became an instant hit with those wanting a notoriously tough dog with sleek, good looks. The Dogo Argentino belongs to the AKC working group and to the FCI Group 2 Pinscher and Schnauzer, Molossoid and Swiss Mountain and Cattle Dogs, in the Mastiff subtype. Care and upkeep. Striking a pose. Dogo Argentino. The Dogo is not a dog for dog parks because of its potentially aggressive nature. It needs frequent exercise, which can be aided by a secure fenced yard in which to run and supplemented with leash walks and other activities such as games, agility, obedience, scent work, and other activities that engage both its mind and body. The Dogo enjoys playing with a ball or a disc, but it may not retrieve them. Coat care is minimal, consisting of occasional bathing and brushing. It does not like extreme cold or extreme heat. As a breed susceptible to canine hip dysplasia, puppies should be raised on a large breed puppy food, which slows the rate of growth and lessens the risk of dysplasia. Brainstem auditory evoked response, BARE.